Hi everyone. Today we're going to discuss how to design a good figure for your publication. When you read the literature, every paper you read, somehow they will have the figure in it. Because figure is very important that help you to write your content. Not only that, it will also help you to write your discussion. Therefore, how to present a good figure is a key to generate a good paper. When do you need to use a figure in your paper? When you need a figure, it is because you want to emphasize the findings. So normally, for example, you have a positive finding or something important. You want to emphasize the finding. You want to attract readers' attention. So in other case, you might use a table. When do you need a table? So for example, you have a lot of data. Some are positive, some are negative, some have a trend of the difference. Others just no difference at all. So there's no need for you to generate a figure. In this case, you will produce a table. When you should really concentrate in writing in your text, particularly in your results section. It is about the details when you explain the meaning of the results. And when you have more explanations, that's where you need to use the text. So what can a figure contain? So when you generate a figure, the first type of data will be the images. For example, you have treatment group, you have a control group. You select representative individual photos, then put into as a figure. This can be MRI, magnetic resonance imaging, produced image. For example, brain image, liver or joints, etc. Then you have the control group, you have a disease group, or you have a treatment group. Then you produce the images. Or you can have the histological tissue sections. For example, the sections of the brain tissue or liver tissue. You take a photo, then this can be used as an image. Other cases like cells, biological cells, you take a photo. For the Western blot is to detect amount of protein expression. This is also an image from a gel then exposed to the X-ray film, then you take a photo from there. Or you want to detect a gene expression, for example, PCR gel image. Those all can be used in the figure. The part 2 is about the histograms. The previous image or photo just represent the whole data by using one sample. Now this is average. You make a histograms. You can compare between the groups. Or you can make a line joins and that demonstrate within group variation. For example, the baseline after 1 week treatment, 2 weeks of treatment. So therefore you put a within group like a line plot. Or you can use line plot to do the dose response data or time effect data because you have multiple points. The third type is called the scattered plot. You can plot the control group and the versus treatment group. In the plot, you will show the individual data. Because you show the individual data, you saw the data distribution. Others, such as you can use box plot to put that into your figure. So figure types, or the types of figures, will really depend on the aim of your presentation. What do you want to present? For example, if you want to present your results in terms of increase or decrease, if there is association, so that's a type of figure, just the results. Or if you want to present a story using a single figure, then in this case, you will need several experiments to talk about one idea from a different angle to prove it. When you arrange this figure, you can arrange the sub-component in this figure, arrange them in a logic or systematic order to present one story. Or you can design a figure to fill the scientific gap. In this case, you present the results, you identify where is the scientific gap by using a flow chart. So this will be a good presentation. First, let's talk about how to present the difference and association. In the following slides, I will give you a couple of examples. For example, this paper is about a pro-inflammatory cytokines, including interleukin-2, interleukin-6, interleukin-8 levels in schizophrenia patients with motor dysfunction, which is tardive dyskinesia. You can see here, this is the figure. This figure shows the difference. This is patients with tardive dyskinesia. Patients without tardive dyskinesia, this control about their interleukin-2 levels. When you see here, this clearly is a scattered plot, allow you to see individual data, average, then standard deviation for all three groups. So this is a scattered plot. Now on the right side, using those data, you can produce an association plot. You see the increased interleukin-2, then you have increased AIM scores. This is a motor dysfunction scores increase. There is a positive correlation, p-value less than 0.01. This is the first type. So show the difference and association. The unique feature of this figure to aid a flowchart here in a figure. This flowchart is to show the event of sequence. First, you have a scratch wound to the tissue here. After 1 hour, you use either PBS or you use LPS, lipopolysaccharide, in this group, or just the scratch control group. Then here, you add a drug treatment. This is called ibrutinib. As you can see, after 24 hours, there is a growth effect in the LPS treated group, but which is prevented by this drug, ibrutinib. This is a quantification and a summary. Histogram. Now, when you design an experiment to show the time effects, that is very powerful because it does provide a very solid data and a very convincing data. For example, this paper is published by this group of people, Joshua et al., in Journal of Clinical Investigation. The title of this article is Obesity is associated with hypothalamic injury in rodents and humans. Here just one figure. In this figure, they have showed the control group, chow diet. This is the treatment group on high-fat diet. 1 day, 3 days, 7 days, 14 days time effects on top of a high-fat diet. What they have shown here is the number of microglia cells increased with the increased time of high-fat diet. Here's the quantitative plot using a histogram. See the increase after 3 days of high-fat diet. This is significant. And also 7 days and 14 days. Also, they look at the size of the cells. It's bigger. As you can see here at day 7, it's much bigger than this. So this is a histogram. So therefore, this data is very convincing to indicate there's an increased number and size of microglia cells after high-fat diet in the hypothalamus. They have also added 2 correlation plots in this figure. It demonstrates that the high fat mass gain, how much fat mass gain, this is the increase, and here the vertical is the cell number in terms of microglia cells. You can see a positive correlation between the number of cells and the fat mass gain, R value and the P value less than 0.05. Here's about the cell size of microglia cells. Also size increases when you have more fat mass gain. It's clearly a positive correlation. P less than 0.05, R value is 0.52. Here's a paper we published last year. This is related to association between hyperuricemia and the metabolic syndrome in patients suffering from bipolar disorder. Because we want to show the risk for metabolic syndrome, we have used the OR-odds ratio. Based on our data, we saw the age and uricemia, uric acid level both contribute to the risk. Therefore, we use this 3-dimensional plot. As you can see here, this is age less than 50 years, age greater than 50 years. And here you have hyperuricemia. Surely you see this is very high risk to develop metabolic syndrome. If the person is greater than 50 years of age, as well as have hyperuricemia, it's about 13.977 times higher in risk to develop metabolic syndrome compared to the controls.